Okay, this is your final review for your exam. I'll just suggest to you, if there's something that you understand, go ahead and fast forward it. If it's something you need, pause it, rewind it as needed. So that's the beauty of this. Okay, so let's get rolling. Okay, so the first topic is the 9 characteristics of living things. These will be simple multiple choice questions, basically about the definition or something about them. So let's go through them real quick here. First one's reproduction, and reproduction we know is having offspring. Okay, there's 2 different kinds of reproduction. There's asexual and sexual. Okay, and so asexual means without a partner, sexual means you have a partner of some sort, right? Okay, heredity. Heredity is when you pass on your genes to your kids. Okay, so you're passing on genes or traits or DNA to your offspring. It's not that you just have DNA, but that you pass them on to future generations. Response to stimuli is responding to some change to your environment. Okay, it's how living things respond to changes in their environment. If it gets cold, you know, they grow hair. If it's hot, they move away, whatever the case may be. Okay, so it's responding to some change. Okay, genetic code. Genetic code means you have DNA or RNA, okay? Means you possess it, okay? So you gotta have DNA. Homeostasis. Homeostasis is maintain constant levels. So, for example, 98.6 is our body temperature. We maintain that. We maintain fluid level, body level. Cellular organization. Fancy just means every living thing must have at least one cell. Remember, if you have one cell, you're called unicellular. And if you have many, you are multicellular. Okay? Growth and development means you're going to grow and develop. You're going to grow through those stages of development, of maturing. Okay, so it doesn't mean that you change over time differently. It means that you're developing to normal stages of your life cycle. Okay, evolution is different. Evolution is how you living things as a whole can change over time, and that is change over time. Okay, and lastly, metabolism. Metabolism has to do with your energy production and usage. So it means, you know, can you make and or use energy in some way. So you might be eating sugar, you might be breaking it down, you might be producing things, whatever it is. Okay, so there's your 9 characteristics. Number 2, how are living things organized and named, and what language is it? So it's all about classification. So remember that classification was developed by a scientist called Linnaeus. And he basically started grouping living things based on a lot of things. He based them on their characteristics. He based them on where they lived, how they reproduced, you know, things like that. And he started grouping them into groups that made sense, that made it easier to categorize them and talk about them. And then he started naming them. And when he named them, he gave them a 2-word name. The name is called binomial nomenclature. Bi meaning 2, nomial meaning name. You don't have to know that word, but if I use it, you'll know where it is. Binomial nomenclature. Okay, so that's a 2-word name. It was based on a common language and tried to create a common language. The language of origin was Latin, as is a lot of science and medicine. And so they used Latin, and they tried to create this common language of, scientifically naming things so that there's no confusion about what something is. Common names tend to change from place to place and regionally, and scientific name does not. So remember, it's a two-word name. For example, you saw in the bacteria Escherichia coli, which stands for E. coli, is a scientific name. It's got a two-word name in it. Okay? So if you don't see a two-word name, it's not a scientific name. Okay? Remember that there are now 1.75 million living named species on Earth. 1.75 million named species on Earth. Most of them live in the tropics. Some of them are extinct. Some of them are still yet undiscovered, so there might be more. They think there's anywhere between 10 and 100 million that are in existence out there. Okay? But these are the ones that we have named and we have categorized. Of those 1.75 million, they all fall into one of 6 kingdoms: protists, fungus, animal, plant, archaebacteria, and eubacteria. Okay? Remember that the archaebacteria are the oldest. They're 3.5 billion years old. They're the first living things on Earth. Okay? And so these guys live in extreme environments. Okay? And they're chemotrophic, which means they use chemicals for their energy. They're unicellular, and they reproduce asexually. Okay? The next thing, the eubacteria evolved from the archaebacteria about 2.5 billion years ago, and they are not extremophiles. These are the bacteria that cause disease. Okay, so all these diseases we've been looking at the last week or so, all are eubacteria, and they are heterotrophic, which means they get their energy from other places. They get it from us when they cause disease. That's why they cause disease, because they're feeding off you or decaying, things like that. They're also unicellular, and they're asexual. Okay? The next organism to evolve are the protists. They're 1.5 billion years ago. Remember that the protists are like the in-between organisms because they have characteristics of everything. Okay? They live in water, and they're both auto and heterotrophic because there's examples of both. Some are photosynthetic, some eat off other protists and bacteria and viruses. They're both unicellular and multicellular, though I would say there's more multi than uni, but there's both. Okay? And they reproduce sexually or asexually depending on which one. So remember that this is that in-between group, and so they can do both. Okay? Fungi evolved next. They're about 700 million years ago. And the funguses are decayers. So they break down things. So they're mostly heterotrophic. Though there are some autotrophic examples of them. They're mostly multicellular. But there's a few unicellular examples. And they're mostly sexual in their reproductive ways. But again, they do have examples of both. But they tend to be more, on this side of it. Okay? Plants and animals both evolved about 500 million years ago, so about the same time for them, which makes sense. Animals are all heterotrophic, and they're all multicelled, and they're all sexual. And plants are all autotrophic, and they're all multicelled, and they can do both sexual and asexual. So they can do both, but they tend to do a little bit more sexual than asexual. They could do both. Okay? So you can see that the diversity of living organisms in the six kingdoms changes as time goes because things are getting more advanced. They're getting more specialized, right? They're going from prokaryotic to eukaryotic. Okay, there are two basic types of cells or organisms. They are prokaryote and eukaryote. Okay, so prokaryotes are bacteria. So of your six kingdoms, the two that I just talked about, the eubacteria and the archaebacteria, are your prokaryotes, and your eukaryotes are everything else. So that's plants, animals, fungus, and protists. All those are eukaryotes. Okay, when did they evolve? Go back to your six kingdoms, and you can see the prokaryotes are like 3.5 to 2.5 billion years. And the eukaryotes are a little bit newer. They're about 1.5 billion. So they've evolved more recently. Okay? In terms of their cells and their size and their complexity, remember that prokaryotes are small. They're simple. Okay, they're not specialized. They are about like 1 to 10 micrometers. Okay, they're small, simple. They're unicellular, and they're old. Okay, so that's an easy way to remember what the prokaryotes are: small, simple, unicellular, old. The eukaryotes are bigger. So they're like 100 to 1,000 UM. A UM is a micrometer. They're complex because they're multi-celled. So you're going to get these cells that are going to work together and become specialized. The word I used. Okay, they become specialized, and these are newer. Okay, so these are like the newer, evolved version of these guys, right? I think I compared it to the Ford assembly line. Making one car versus making 100 cars in the same amount of time. It's bigger, better, faster, more specialized. Okay? Remember, the big difference between them that you could look at was the organelles in them, and this one has no nucleus. So that's an easy way to look if you have a picture to decide. Okay, 5. Okay, so the prokaryotes. The prokaryotes are bacteria. Okay, so I just gave you some characteristics of them. Bacteria are living, yes. Okay, and so bacteria are small, unicellular, living organisms. Okay, they have organelles in them, they have parts in them, they do different things. Very diverse group of microorganisms. Okay, micro meaning small, organism meaning living. Okay, so we talked about the diversity of the bacteria. We'll talk about the kingdoms in a second, but we know that all bacteria fall into basically one of 3 shapes. Okay, and so here's those shapes. The first one looks like this. It is circular or spherical, and it is called coccus. Okay, and so you saw that in like Staphylococcus and Streptococcus. Okay, the next one is rod-shaped. A lot of times they have a little tail on them. Remember the flagellum, like E. coli, and this is called bacillus. You saw that in Bacillus anthracis, the anthrax one. It clearly had that shape, so that's how they named it as such. Okay, and then the third one is spirillum, and that is the spiral-shaped one. Okay, the spirillum, and so some of them are spiral and they twist and bend and so on. Okay, so even though they're diverse, there's only 3 basic shapes. Okay, 2 kingdoms of bacteria and how they obtain energy. Remember that they are the archaebacteria and the eubacteria. Okay, so remember that the archaebacteria live in extreme environments. So the earth was nasty when they were around. The only thing was around was chemicals in the sun, so they had to use those. So they are chemotrophic and they are autotrophic. Might be called photoautotrophic, photo meaning sun. So here they used chemicals. In the water, you're talking about methane and sulfur and poisons and toxins. Here they're autotrophic, they use the sun, they're photosynthetic. Okay, these don't make you sick, they just live to survive and to reproduce. Okay, so they're very ancient and old, they're archaic. The eubacteria are newer, and these are the ones that cause that disease that we talked about. So these guys are feeding off other things, that makes them heterotrophic. Okay, they're feeding off other things, so you know they're decaying things. They're living off organisms, and they're making things sick. They're causing disease as a result of that. Okay, so they're heterotrophic, feeding off other things. Okay, optimum bacterial growth and how to grow it. So we grew bacteria from the school in different places. Remember, you need a petri dish. You need nutrient agar. Okay, so the petri dish provides a safe place to live, a space. The nutrient agar is going to provide a food source, and the bacteria is then going to be put in an incubator for 2 days, depending on the type of bacteria. And the incubator is going to provide heat, which is going to make that grow a little bit faster. Remember that high heat and low heat will inhibit growth, but the right amount of heat, right around body temperature, or right around 100 degrees, is going to make that bacteria grow faster. Okay? And remember that when they do that, bacteria grow as colonies. Okay, and so you looked at the colonies in the petri dish to see how they grew. Different colonies or different bacteria. When it is reproducing, to produce those colonies, it's reproducing by a process called binary fission. Okay, so binary fission is a fancy word for how bacteria reproduce asexually. Okay, and so basically what you have is you have one bacteria. It's going to copy its DNA. That's called DNA replication. So it's going to copy DNA right here. You can see 2 copies of the same thing. So that's how bacteria are identical to each other. Here it's going to stretch out or elongate. And so it's starting to build more material, get bigger. It's starting to pinch off right here. Eventually it's going to pop right apart right there and split into 2 and separate. And then from here they're 2 little babies, and so they've got to grow before they can do their job. Okay, so that's basically how bacteria reproduce, how binary fission works. Remember that binary fission doubles every generation, and a growth curve of it is going to look like this. It's going to be exponential growth because you're doubling every generation. Okay, E. coli. Again, is the bacteria that's in your digestive system, in your bowels, in your poop. Okay, very common. It's obviously a rod-shaped bacteria, bacillus. Couple of important structures to note. Number 1, this is the DNA. Notice it does not have a nucleus around it, which makes it a prokaryote. Okay. The other things are, normal structures that we have. So don't need to really address those, but let's address a few specific ones. Remember, these are the pili, and those are the little knobs that help it to stick to stuff. This tail is the flagellum, and that lets it move. And then notice that there's 1, 2, and 3 layers of protection around the E. coli. The first one is the cell membrane. The second one is the cell wall. And the third one, this big outer red part, is called the endospore. And so again, in terms of our food, bacteria like E. coli that have an endospore are dangerous to us because you have to heat them up higher than normal to kill the endospore, to kill the bacteria. So this is one of the type of bacteria that cause food poisoning. Okay, antibiotics versus vaccines. Remember that. An antibiotic you take you take after you're sick. Okay, and so you take this to help you feel better. You've been infected. What this does is this stops bacteria from reproducing. Okay, it stops binary fission from happening. So that will let your body catch up, and eventually you'll fight it off. That's why you don't feel good right away. It takes you a couple of days. Okay, vaccines you take before you get sick, and they prevent sickness and illness. In some cases, it totally prevents it. In others, it maybe protects you from getting it less. Okay? What these guys give you is they give you a dead or a weakened strain of whatever it is to get your body a snapshot so that your body will recognize it and it'll be able to fight it off. In some cases, it will prevent it totally. Okay, so vaccines are given to you to prevent getting sick. Okay, the bacteria that cause disease, the different diseases we talked about. So remember, E. coli, salmonella, and botulism are all causes of food poison. Legionella, which you'll just hopefully remember, Legionnaire's disease from Flint, and cholera, and Salmonella typhi. These are all waterborne. So they have very similar symptoms to each other. They're very similar. Okay. Rickettsia, you probably don't remember that one. Spotted fever. Y. pestis, you should remember. That's the plague. And so this was caused by ticks. This is caused by ticks, and this is caused by rats. So this is animals. Okay, I would know the plague. Okay, Bacillus anthracis is anthrax. Clostridium tetani is tetanus. This was found in soil and dirt. Step on a rusty nail. The soil from your ground. So those things are passed on through the soil. And lastly, strep and staph cause many, many diseases collectively that are common: meningitis, mono, pneumonia, MRSA. The list is on. There's 100. Toxic shock. These are all person to person. Okay, so we can spread things from person to person. Okay, we're going to cross out 15 through 19, and we're going to go into number 20. Okay, so cells. Why is it important for cells to be small? Remember that we compared surface area and volume. Surface area is the area around something, right? It's exterior surfaces. And then its volume includes how much space it has inside, okay? So we compared those two things, and we found that the smaller the cells have bigger surface area to volume ratio. And that meant that it was really beneficial to be small if you're a cell, because you can move materials in and out and throughout the cell much, much faster, okay? Not to mention other things like being able to reproduce faster and being able to have a shorter life cycle and be simpler and work together, and protective things. If one dies out, you've got a lot more left. So there was a lot of reasons for cells to be small. One that we talked specifically about was surface area to volume ratio. And remember, you just take surface area divided by volume. Okay, so the cell membrane. Remember the cell membrane? lets stuff in and out, so it controls how stuff moves in and out of the cell. Here's the parts of the cell membrane. Remember, the cell membrane is predominantly made of fat. Okay, it's made of these things called phospholipid. And the phospholipid is a head with two tails. Okay, so you can see the cell membrane is made of phospholipids that are stuck together and on each side. Okay, some different structures in the cell membrane is a glycolipid. And so the glycolipid is going to play a role in communication. It's going to communicate with other cells or receive information about other cells. So, it's just going to communicate. Okay. C is pointing to a glycoprotein. D is a carbohydrate chain that is stuck to that. I'll just leave it. This whole thing, C and D collectively, is a glycoprotein, and this is more of an ID tag. So this is going to send information about this cell. It's going to tell other cells about it. And so these two things are really communicating with other cells. Okay? This is the outside of the cell. This is the inside. You can tell that because the glycolipids and proteins are sticking out. Okay? G is a channel. Remember that the channels let things in and out based on size, shape, and recognition. Okay, we got to let the right size things, the right shape things in, and we got to recognize it that it's safe to let in. All right. F is just a membrane protein. This happens to be an internal membrane protein, but and it's got a lot of funny names, integral protein, and so on and so forth. Okay. That could be enzymes and other things. Okay. Lastly, this one is a cholesterol. And cholesterol is fat, but it could also be a hormone, so it could be doing some communication things. So that's the parts of the cell membrane. Okay, so we talked about, with the cell membrane, we talked about diffusion and osmosis. Remember, diffusion is movement of materials, movement of stuff from an area of high concentration to an area of low concentration. All right, so remember that just means that stuff is going to move from a high concentration to a low concentration, so that stuff is going to spread out and diffuse to an area. Okay, it could also mean it's going to move across membranes from one side to another. That's the same thing. Remember that this does not requires no energy. Okay, so there's no energy involved with this at all. Osmosis is just a fancy word for diffusion of water through a membrane. Okay, and so, you add cells that we talked about, and you put stuff outside them, and of course they're going to move in because there's none inside. They're going to try to make that thing where everything's the same. That is called equilibrium. All right. Again, it does not require any energy as long as these are small enough to fit through. That size and that shape part of the cell membrane is going to let them move through. Okay. And again, things always move from high to low. Now, we had three specific solutions that cause changes to cells because of diffusion and osmosis. Okay, the first one was a hypotonic. Remember, a hypo means below strength. Okay, and so that meant that the solution is below strength, which means that it's mostly water. Okay, and so that water is going to move into the cell, and it's going to make it bigger. The next type of solution is a hypertonic solution. A hypertonic solution is above strength. And so that means that it's really strong. Gee, I spelled that wrong, whatever. It's above strength. It's really strong, which means it's got very little water in it, and it's got a lot of other stuff in it, like sugar, salt, stuff like that. So this cell in a hypertonic solution, the water is going to rush out of that cell, out of this plant cell too, and it's going to try to make the stuff around it more watery, right? And so the cell is going to shrink or shrivel, same thing. Okay? And then lastly, in an isotonic solution, everything is the same. So you get the same concentration in and out, and so equal parts will move in and out of that cell. Okay? Notice in the plant cell has that extra cell wall around it, and so it's not affected as much by solutions as animal cells because they can burst, whereas this won't. Okay, looks like I skipped 4D, and so let's look at the two types of cells. So in terms of prokaryotes versus eukaryotes, remember that prokaryotes don't have that nucleus. So as you look back at, where did my picture go? You look at this picture, here is a E. coli with no nucleus, okay? Looking at these two cells, you can tell that they are eukaryotes because they have a nucleus right here, okay? So definitely we're looking for that. Now, comparing plant and animal cells, we're looking for three key things, okay? Number one, we're looking at, does it have a water vacuole? Right here, okay? So this one has a vacuole, this one does not. They have smaller vacuoles, which are for storage of food and stuff. This is all for water and sugar. So this is really big. So number one is the vacuole, the water vacuole. Okay. Number two, we're looking for chloroplasts. Chloroplasts are the organelles that do photosynthesis. So here's one, here's one, here's one. They're not drawn real well. Remember that it kind of looks like a mitochondria, but it's got the little stacky guys inside. Okay, so two is the chloroplast. All right, and animals don't have those because they don't do photosynthesis. And then lastly, number three, here on the outside we see a layer and a layer, whereas here we only just see one thin membrane. So number three is the cell wall that the plant has, and the animals do not have that. Okay? Everything else is basically the same. Okay, the organelles, make sure that you know the function of them and or a picture. I know there's a picture for sure. You should all know their parts. I'm going to put them in one word to help you. The nucleus is the boss. The cytoplasm is the jelly. The chloroplast makes the food. The mitochondria makes the power. Ribosomes make protein. The ER is for shipping. The Golgi is for packaging. The cell wall is for protection. The cell membrane lets stuff in and out. The vacuole is for storage. And the lysosomes are for breaking down waste. Okay, so they are going to get rid of this stuff that is stored. All right. Where is it there? Okay, now, just as far as finding these things, I'd be able to find them in pictures. So I'll just draw a line to them here. Okay, the nucleus is here. This is the nucleolus, this is the nuclear membrane, this whole thing is the nucleus, okay? Not going to make you differentiate that. That's the nucleus. That's where our DNA is. That's what makes these eukaryotes and not prokaryotes. Okay, the cytoplasm is the stuff in the cell, the jelly that fills it, the area, the space. Okay, the chloroplast is over here. Mitochondria looks got the little wavy lines in it right here, this thing. Ribosomes are the little dots. There's ribosomes here, and here, and here, and here, and here. And also we know that there's a lot of ribosomes localized to where the rough ER is. So that's where your ribosomes are. They're the little tiny dots. Okay, the Golgi and the ER look a lot alike. So I sometimes will accept them interchange. There's your Golgi. Okay, cell wall is right here. Cell membrane is right here. And then also the plant has a cell membrane too. Vacuoles, remember that there's regular vacuoles and there's a water vacuole. And then lastly, lysosomes. Lysosomes and vacuoles look a lot alike, so this could be a lysosome, that could be a lysosome, those could all be lysosomes or vacuoles, here. Okay, so they look a lot alike. All right, I think that's everything. Make sure that you are asking yourself questions and studying appropriately. Hope this helped you. Good luck. Bye.